IB Bio Animal Physiology Topic 11, Part 5, Movement C, will have its focus on the biochemistry of sarcomere contraction. The movie is not long, but it's detailed. Pay close attention. The essential idea is the roles of the musculoskeletal system are movement, support, and protection. Here is the outline of all the available movies for Topic 11. Use this outline to choose the movie you need for review. This movie is focused here. Here are the first two IB syllabus statements of this movie. The contraction of skeletal muscle is achieved by the sliding of actin and myosin filaments. ATP hydrolysis and cross-bridge formation are necessary for the filaments to slide. You've seen both of these statements in Part 4, Movement B, but they provide good foundation as we step into the biochemical regulation of contraction. Within muscle fibers, actin and myosin filaments lie parallel to one another. The shortening of a muscle fiber comes as actin slides past the myosin. Myosin heads, seen here, form cross bridges that bind the actin and pull the actin to the middle of the sarcomere. ATP is required as the myosin heads hydrolyze ATP. Here is a list of muscle structures from the largest to the smallest. I introduced this ordering of structures in the last movie, and you should be comfortable with the first 7 terms. I've brought this slide back to say that this movie will introduce tropomyosin and troponin as regulatory proteins. This IB syllabus statement should be under your control, but as I wind my way toward the high detail of sarcomere contraction, let me review just a bit. Describe the structure of striated muscle fibers, including the myofibrils with light and dark bands, mitochondria, the sarcoplasmic reticulum, nuclei, and the sarcolemma. The sarcolemma is the plasma membrane of the muscle cell. A muscle fiber is composed of many myofibrils, which are in turn composed of overlapping actin and myosin protein myofilaments. The alternating overlap, non-overlap, overlap, seen as striations, are a hint to the structure of a sarcomere. A sarcomere, seen here, is the functional unit of a muscle cell. It is the sarcomere that contracts. Here is a close-up image of a muscle fiber. Within the cell membrane, the sarcolemma of a fiber, are numerous myofibrils, and these are composed of filaments, actin and myosin, and they're just seen in cross-section. You can see a nucleus, one of many, as well as numerous mitochondria. Numerous mitochondria should make sense to you because muscles spend a lot of ATP to contract. The specialized endoplasmic reticulum can be seen here. It is known as sarcoplasmic reticulum. And T-tubules connect the surface of the cell to deep within the cell to the sarcoplasmic reticulum. The sarcoplasmic reticulum stores calcium ions, and when a muscle fiber is stimulated by a nervous impulse, the sarcoplasmic reticulum releases calcium. More on this later in the movie. Pay close attention. Even more close up on a muscle fiber, here is the cell membrane, the sarcolemma, and here is the sarcoplasmic reticulum, the specialized endoplasmic reticulum. This image better illustrates the actin and myosin protein filaments. The thick protein filament is the myosin. The thin filament is the actin. The image nicely displays a sarcomere. The sarcomere is the functional unit of a muscle cell. The sarcomere, defined from Z-line to Z-line, is the structure that shortens, contracts. The thin actin filaments slide past the myosin filaments to effect shortening. Myosin cross-bridges bind to the actin, hydrolyze ATP, change shape, and move the actin to the center of the sarcomere. Once again, a muscle fiber with myofibrils and sarcomeres. Take some time to study this slide. I like how it displays the relationship of the sarcoplasmic reticulum with the myofibrils. Remember that sarcoplasmic reticulum stores calcium ions when a muscle cell is at rest. You can see numerous mitochondria that provide ATP for contraction. Let's look closely at this myofibril that displays a sarcomere. The thick myosin filaments can be seen here. The thin actin filaments overlap the myosin filaments in some regions of the sarcomere. Upon contraction, the myosin heads form cross bridges binding the actin and pulling the actin to the center of the sarcomere, reducing the length of the H zone. Keep in mind that muscle fibers are three-dimensional and that these myosin heads surround the myosin filament in all directions. Here's the next IB syllabus statement that drives us forward. Explain how skeletal muscle contracts, including the release of calcium ions from the sarcoplasmic reticulum, the formation of cross bridges, the slotting of actin and myosin filaments, and the use of ATP to move the myosin heads. Describe the roles of regulatory proteins, tropomyosin and troponin. Here's a nice image providing a three-dimensional look at the sarcomere. You can see the thin actin filaments anchored to the Z-line. It's a protein structure, the Z-line. You can see the thick myosin filaments with their cross bridges. Here is an isolated myosin filament with the myosin heads clearly displayed. What's nice about this image is that you can now imagine how a sarcomere shortens, how it contracts. The sarcomere shortens when the myosin cross bridges bind to the actin and pull the actin to the middle of the sarcomere. The action of contraction requires ATP. Stay tuned. When calcium is released from the sarcoplasmic reticulum, the myosin heads bind to the actin, forming cross bridges. The myosin heads hydrolyze ATP, they change shape, and pull the actin to the center of the sarcomere. This reduces the length of the H zone as well as the lengths of the I bands. Here is a micrograph showing part of a sarcomere. This is the H zone. The thick myosin filaments can be seen with their myosin heads. The actin is here, ready to be pulled by the myosin cross bridges to the center of the sarcomere. But the actin filaments are not alone. Actin has two other proteins associated with it, tropomyosin and troponin. Tropomyosin and troponin are regulatory proteins in the contraction of a muscle fiber. Here is a diagram of actin in orange with both tropomyosin and troponin proteins. Actin is closely associated with tropomyosin and troponin. Notice the binding sites on the actin, but they appear blocked, unopened. The binding sites are the sites to which the myosin heads bind to form cross bridges. At rest, when the sarcoplasmic reticulum has not released calcium, the binding sites are blocked. As you see here, they're blocked by the tropomyosin. When a nerve sends an impulse to a muscle, calcium ions are released from the sarcoplasmic reticulum. The arrival of calcium ions results in the opening of the binding sites on the actin. Stay tuned. Here is a micrograph and a diagram of a myosin filament displaying the myosin heads. These are the cross bridges. The images nicely display the three-dimensional nature of the myosin filament. The myosin heads are ATPases. The heads are enzymatic. In other words, the myosin heads bind and break ATP. They hydrolyze ATP. As they hydrolyze ATP, they change shape, pulling the actin. This schematic diagram helps us understand the biochemistry of sarcomere contraction. We can see the actin, the tropomyosin, and the troponin. We can see the myosin and the myosin head ready to form a cross bridge. At rest, the binding site on the actin is blocked by the tropomyosin. The myosin head cannot bind. At rest, the sarcomere is not contracting. Upon nervous stimulation, calcium ions are released by the sarcoplasmic reticulum into the cytoplasm of the muscle fiber. The calcium ions bind to the troponin. The troponin is in contact with the tropomyosin. Once the calcium ions bind to the tropomyosin undergoes a conformational shape change, opening the binding site for the myosin head. In this diagram, you can see how the calcium ion is bound to the troponin, resulting in a conformational shape change in the tropomyosin, opening the binding site that's on the actin. The myosin head can now bind to the actin, and under the influence of ATP, the myosin head will change shape, and the ATP-driven shape change of the myosin head pulls the actin toward the center of the sarcomere. Here is the muscle fiber at rest. Without calcium, the binding sites on the actin are blocked by the tropomyosin, a regulatory protein. With nervous stimulation, the sarcoplasmic reticulum releases calcium. The arrival of the calcium ions bind to the troponin, results in a change in the position of the tropomyosin. This opens the binding sites on the actin. Once again, but a different image. We have the actin, the tropomyosin, and the troponin. Here is the myosin with a head that binds ATP and hydrolyzes ATP. You can see how the myosin heads might bind to the actin if the binding sites on the actin were open. Now at rest, the binding sites on the actin are blocked by tropomyosin. The position of the troponin-tropomyosin complex can be changed under the influence of calcium. Here is the actin with the troponin-tropomyosin complex, and with the arrival of calcium ions released from the sarcoplasmic reticulum, the calcium ions bind to the troponin, causing a conformational shape change in the tropomyosin. The tropomyosin shifts position, revealing the binding sites on the actin. The myosin heads bind to the actin, hydrolyze ATP, and change shape, pulling the actin to the middle of the sarcomere. Here are two IB syllabus statements that should feel relatively comfortable at this point. ATP hydrolysis and cross-bridge formation are necessary for the filaments to slide. Calcium ions and the proteins tropomyosin and troponin control muscle contractions. Here is a diagram that simply shows how the myosin head binds to the actin, hydrolyzes ATP, changes shape, and pulls the actin to the middle of the sarcomere. The myosin head binds to the actin. ATP is hydrolyzed and ADP and P are released. The head pulls the actin and the Z line moves in. More ATP binds to the myosin head, resetting the position of the myosin head. The head binds to the actin, hydrolyzes ATP, and pulls the actin to the middle of the sarcomere, shortening the sarcomere. And the process is repeated over and over again. Zooming out a bit, you can see the myosin heads, and you now know how they bind to the actin and undergo a conformational shape change under the influence of ATP. The myosin heads pull the actin to the center of the sarcomere. The I band and the H zone disappear as the myofilaments slide past one another. Here's another image of a sarcomere that nicely displays the myosin cross bridges as they bind the actin once calcium ions arrive from the sarcoplasmic reticulum. The myosin heads hydrolyze ATP to pull the actin filaments past the myosin toward the center of the sarcomere. Here is a diagram with the entire process starting at 1. The myosin head has bound ATP, but the actin binding sites are blocked by the tropomyosin. Calcium arrives from the sarcoplasmic reticulum upon nervous stimulation. The binding sites on the actin are now open. The myosin head binds to the actin. ATP is hydrolyzed. The myosin head undergoes a conformational shape change, pulling the actin toward the center of the sarcomere. The arrival of more ATP releases the myosin head from the actin. The myosin head is now ready to bind and pull again. So here is a diagram that reviews the big picture and small. An impulse arrives at the neuromuscular junction. Neurotransmitters depolarize the membrane of the muscle cell. The depolarization sweeps across the muscle cell membrane, moving deep into the cell through the T-tubules. The T-tubules have appeared in other images I've displayed, but I've not spoken about their role until now. The T-tubules make contact with the sarcoplasmic reticulum, seen here, and this results in the release of calcium ions. The calcium ions bind to the troponin, which is associated with the tropomyosin and the actin. The arrival of calcium results in the opening of the actin binding sites to which the myosin heads can bind. The myosin head hydrolyzes ATP and pulls the actin to the center of the sarcomere. The calcium is actively moved back into the sarcoplasmic reticulum as the muscle begins to rest. New ATP would bind to the myosin head to ready the myosin for the next impulse. So are you comfortable with this statement? Calcium ions and the proteins tropomyosin and troponin control muscle contractions. Could you relate this statement to the sarcoplasmic reticulum? I'll let you study this slide to review the syllabus statement. Calcium ions and the proteins tropomyosin and troponin control muscle contractions. In this diagram, we can see a neuron making contact with a muscle fiber. Neurotransmitters are released by the neuron. The neurotransmitters cause the muscle cell membrane to depolarize, and that causes the release of calcium ions from the sarcoplasmic reticulum. The sarcomeres within the muscle fiber will contract. In this slide, we can see 3 muscle fibers with striations and multiple nuclei within each cell. These elements of the diagram should be clear to you. As well, we can see neuromuscular junctions. You can see the schematic sarcomere within a fiber. The overlap of actin and myosin within the sarcomere is responsible for the striations. Neural impulses cause muscles to contract. Neural impulses cause sarcomeres to contract. You should be able to explain the contraction of sarcomeres to the extent of describing the roles of calcium, troponin, and tropomyosin. Muscle fiber contractions are controlled from the central nervous system by neurons that make synaptic connections at neuromuscular junctions. And that brings us to the end. IB Bio, Animal Physiology, Topic 11, Part 5, Movement C.